donated by and large pro bono. Experiments are done at cost. We have a huge amount of philanthropic funding from these organizations here, plus more support, as I'll come on to detail. It's a truly international effort. You can see the sun never sets on the COVID Moonshot project. There's always somebody working on it somewhere. We have a unique constitution. It's an open science project. It's a global, extremely flexible team, which has been vital working during a pandemic. We have hundreds of years of big pharma experience between us, and that's also led to access to BSL-3 antiviral and in vivo testing facilities. Okay, what are we trying to do? From the very start, we're trying to create a drug, and we're trying to create the right drug. We know so much more about COVID now. You have an early phase, where the viral load gets very high. That leads to mild to moderate illness. And then you get an immune overreaction. That usually kicks in around about day 7. And for people who suffer that can lead to severe illness and obviously mortality in some cases. What we are trying to do is work here in the oral antiviral space, where people are either early symptomatic or even pre-symptomatic. We need to be able to reach the non-vaccinated population, both those people who are at risk, the non-responsive, and of course the vaccine hesitant who can still get ill. If we can knock this down, we will reduce both hospitalization, mortality, of course also long COVID. It's an open science project. That means you can dial in and see what our plans are, what our results are. This is the website that shows each plan has a little thumbnail of a representative compound. You can then drill further. So say for this compound, what were we planning? You can see here are 4 compounds planned, a little rationale, and it's got who suggested it and date stamps for all of it. And then you can actually look in and say, well, okay, this compound, what are its calculated properties, but more importantly, what's its actual measured properties, and when was it ordered, when was it made, when was it shipped, when was it tested? You can also look across and say, okay, that looks like an interesting compound, and you can look on the Diamond website using Fragalysis, and you can pull up the crystal structures with the electron density maps, so you can see the detail to actually interpret those crystal structures. And we have over 390 crystal structures now available. So what does open science mean in terms of actual outcomes? We are aiming for equitable global access. So that's a direct to generic strategy. As I've just shown you, all of our plans and data is reported and date stamped and accessible, and you get more detail on CDD. We're taking an active strategy to try and maximize the amount of public good we do. That has some important positive consequences. We have open access to people to contribute to designs. You can all contribute if you want to. We have very fast collaborations. We have no CDAs, no MTAs. That's turned into access for BSL-3 facilities and animal models. And compounds are available for our collaborators in Enamine's catalog. You can just order them and get samples. Of course, under those circumstances, we have to post warnings about the fact that these compounds have not been tested in terms of toxicology, so you shouldn't be using them therapeutically. Okay, what's our target product profile? All the usual things, so primary target potency, cellular potency. We'll talk a little bit about our PK/PD approach, appropriate coronavirus spectrum. We want oral drugs, we need them to be soluble, we need them to generate exposure, and the usual clean safety profile, particularly looking at protease selectivity, obviously. Okay, there are challenges in the assays. In terms of animal models, the ferret is one of the default respiratory models, but it's a very complex model to use. Hamsters get a very severe disease and are very strong metabolizers. If you're looking in mouse, you can either look at the ACE2 humanized mouse, or you can look at the modified murine coronavirus with wild type mice. Both of those are a compromise. So the pragmatic option is look at predicted human semen cover over the EC90 in a relevant cell line. But what cell line is relevant? All the cell lines you might think of are in some ways compromised. VEROE6 is the workhorse, but it's an immortalized monkey kidney cell line. It's interferon deficient and it's PGP overexpressing. CALU3 and A549 are both lung immortal cell lines with their own challenges. Replicon assay hasn't been available until only very recently, and certainly not when we started. You might want to test in pneumocytes, but they're not accessible for screening. But even worse is actually just getting access to a BSL-3 facility. We have been able to do that. We have multiple collaborators, the IIBR in Tel Aviv, University of Oxford, and recently the University of Louvain have all been able to provide access to, in terms of testing, to their BSL-3 facilities. But the enzyme assays are also not trivial. MPro is autocatalytic in its own destruction. It forms a dimer with one active site. So we have two assays running, a FRET assay at the Weizmann Institute and a rapid fire assay in Oxford. Okay, the key elements of the COVID Moonshot project. We have a fragment-based strategy aimed at safe oral compounds. We have a lot of AI accelerated chemistry by using a lot of reagent diversity, route selection, and we've used massive scale FEP for calculations. And I've just talked about our biological screening capability. We think with the strategy, the compounds, and the assays, we're aiming to generate 5 NCE candidates for preclinical evaluation. Okay, our assay cascade is configured to be very stripped down and focus on the problems in hand. Primary enzyme potency, rodent cassette PK, usually as racemates, we then resynthesize and resolve. I'll talk about that later on. Antiviral assays in parallel. We had been focused on trying to get into a mouse efficacy model, but now we've switched over, generally looking at rat, oral and IV PK, then human and rat heps and mix to generate a human PK prediction package. And in parallel, selected examples for safety testing. Okay, let's get into the medchem. That fragment screen was amazingly successful. Here you can see multiple fragments binding at the P1 site, contacting the histidine at the bottom of the pocket and also this backbone amide. You can combine that when you see the data from the P2 fragments bound here, where all of them are dumping halogens into the bottom of that P2 site. And one of our very first designs from Trifon Zarganis Tsitsikas at the University of Oxford was just to say, well, look, we can see these P1 fragments, we can see these P2 fragments, let's just couple them up by eye and make compounds like this. And that was an amazingly successful strategy. So here's exactly that done. That compound made is 24 micromolar from a standing start. Okay, what we do when we think about design is we're aiming for small, efficient molecules that should avoid off-target effects, reduce permeability and metabolic risks. Importantly, try and keep well within the substrate envelope to minimize resistance risks. Keep the compound simple. In turn, that will reduce cost of development and cost of goods, which means we go faster, and faster in the end means equitable access. We've decided to avoid peptide mimetics. That just presents a different development and toxicity risk profile. I think we'll need both in the end. What about covalency? Our strategy is to make the compounds potent and selective first and add a covalent warhead if we need it. That means we're focusing on selective, efficient ligands rather than hot warheads. I'm not going to talk about the pan-coronavirus spectrum. That's not one of our primary goals. Okay, how do we optimize this compound? I'm going to show you the actual design ideas that led to the compounds along the bottom here. So this compound was rapidly evolved by annihilation of the extra ring here, where you can see there's the original, all of these are crystal structures. So you can see the crystal structure, the pyridine, the isoquinoline overlays beautifully, as does the rest of the molecule. We get a very significant step up in potency. So this was a huge step forward. And that came out of the enamine real collection. We only made 2 compounds out of that design, but it was a great couple of compounds. This is a really interesting compound. It was made from a pharmacophore search of enamine real. You can see this replacing the isoquinoline, this annihilating on the side of the 3-chlorophenolacetamide. And you can see how, again, that comes around beautifully in all overlays. The problem with this compound is, although it's active in the fluorescence assay, it's not active in the rapid fire. And we had a lot of soul searching about this compound, and we think it is actually a mirage in terms of potency. But what it did lead to was, as we were working on this compound, Vladas at UCB proposed taking this improvement of the isoquinoline and just reacting it with the best of the acids that we thought we'd found here. And that compound does reproduce. So now we're down at 190 nanomolar as a single enantiomer, and that's a fourfold step up from where we were here. And that was just expansion around this hit. Okay, you can build potency further. So here's the benzopyran lead. We did the standard medicinal chemistry things. We worked around looking for small groups that we could put around the molecule. This was one of them, trying to bias this axial conformation by adding in the extra methyl. Now that doesn't work. Okay, none of these small substituents make any significant improvement. But what it did do was inspire another design by Peter Kenny, saying, well, okay, we know we can put groups in here. Can we reach across into the S1' subsite? And a good test compound will be the methoxy, as I've indicated here. And that does give you a very significant improvement in potency. So that's more than twofold. And in terms of lipophilic ligand deficiency, it's definitely a step up. But potency is clearly not everything. We know that. So you profile the compounds more widely. And here you can see very reassuringly, we're getting real antiviral activity now, and it's improved in the methoxy compound. We also have oral exposure. This compound, unfortunately, we weren't able to perform the IV leg because of the really marginal solubility. So we weren't able to formulate it. But with the methoxy analog here with better solubility, you can get quite respectable oral half-lives, and you can get some significant bioavailability, both with reasonable amounts of free drug. Okay, at this point in the project, we were still focused on trying to get into a mouse efficacy model. And so here's just what the pharmacokinetic profiles look like against the two different cell lines. And although we do get some cover, and if you step the dose right up, you do get cover for a couple of hours over at least the Carolu-3 cell line, this isn't sufficient to go into a mouse efficacy model. So we want to push this forward. Simultaneously, of course, good medicinal chemistry continues. We'd also searched around what else can you put round into the P2 site. And Ralph put forward this design, a very systematic exploration of the SAR. This compound came through. It's a small step forward. Again, if you combine this learning with what we found out about the methoxybenzopyran, it doesn't appear to make a significant amount of difference. But again, potency is not everything. If we look at that in comparison to the simple chloro analog, we do make a step forward in terms of antiviral efficacy. Looks like we make a step forward in terms of potency, solubility. In terms of human hepatocyte clearance, it's significantly better. And you also make an improvement in terms of both bioavailability and the free drug goes up. So when you look at all of those factors, the potency and the predicted human clearance and the exposure, you've got to ask the question, have we moved our compound on enough in terms of predicted human dose? So we started doing human dose predictions with this compound. This is all the data gathered together. Here's the profile, here's the crystal structure. And you can see here our predicted human profile. Now, the real weakness of the compound is, although it has got a half-life of over an hour from IV dosing and almost 3 hours from oral dosing, the clearance is still too high. And what that leads to is not a great predicted human dose. Now, the range, 300 to 3,000 mg per kg, our best guess is around about 1 gram, 3 times a day. If it was the bottom end, that would be great. But if it's a top end, clearly unacceptable. And that's going to require dosing to a second species to get precision on that estimate. So you ask yourself, is the benzopyran actually the problem? And we've done a very thorough scan around changing the heterocycles, you see X1, X2, X3, looking for what we can learn about where do we get potency, and also where could we reach out more in terms of these compounds. We've actually found you can put more polarity into this center. So both the SO2 and actually with the carbonyl here, so making the amides, those both work. But the non-basic tetrahydroisoquinolines look really promising. Okay, we get a huge number of designs into the project, and I want to talk about concepts of over-design and under-design. So when people over-design compounds, that's usually based on overestimating their ability to predict potency, solubility, clearance, really complicated factors. And they often come in with molecules that are too complex, which are then slow to make. But also there's under-design, not taking account of the SAR we've already got, making molecules with poor properties, and making insufficiently diverse compounds. And that just leads to failure. Okay, this is a so-called kind of Goldilocks design that Matt put in. Looking at the substitution on the tetrahydroisoquinolines, just making 5 compounds. So the methyl, the sulfonamide, the urea, the acetal, and the unsubstituted urea. This was a spectacularly successful little set of compounds. So all of them, with the exception of this, are really potent. We wanted to push that forward. So can we reach out to glycine 189, make a little library around here, all constrained by physical properties? Yes, we can. Obviously, it works really well. So the sulfonyl ureas are again potent, and we've pushed down the lipophilicity. We can get more potency. Somewhat interestingly, our strategy of trying to put on substitutions here that would reduce the basicity of this nitrogen by adding electron withdrawing groups here looks like it's worked really well and has generated some diversity in terms of the structures. That was requested, took about a month to turn that work around. We followed that up with a second generation of sulfonamides, so making more sulfonamides, of which one of the best is shown here. So now that is the potency as the racemate, so the single enantiomer potency is about half that. Gratifyingly, we have changed the clearance profile, so the compound in rat is cleared more slowly, has a lower volume, and therefore we've pushed the half-life up. In terms of our design idea of reaching out to glycine 189, that hasn't worked in either of these cases. The compounds have both folded back. In this case, up against the protein. In this case, it looks like it's forming an internal intramolecular hydrogen bond, which presumably is reducing the desulfation penalty, which is where we think we're getting some of the potency in this compound. Okay, again, just because it's potent and we've got clearance, that's not enough. How does that compound look when we profile it across our cell panel? So here's the sulfonamide in either A549s, Calu3, Vero-E6. Each point is a compound in a cell line, so these connectors show the same compound in different cells. You can learn some things here. So the Calu3 appears to be more sensitive, a bit more than the Vero-E6. The A549s are a bit demanding. But here are 2 of our leading compounds showing their profile against a Pfizer compound that we're about to hear more about. And our most potent sulfonamides are getting into roughly the same ballpark as this Pfizer development compound. So that's amazingly exciting. Here's where we are from a cold start in March 2020 to August this year. We aren't quite where we want to be in terms of our PK/PD exposure over cell potency, but in most of the rest of the properties, we are achieving what we want. I haven't been able to tell you a lot of the stories that the COVID Moonshot has generated because of time. We know a lot about SAR around the isoquinoline. We've done a huge amount of work looking for isoquinoline replacements. We've tried to reach into P1 prime, looking at that's been strongly constrained by synthesis. We've done a lot of AI-supported compound and route design. I'll tease you with the spiro compounds and their pan-corona activity, and then rapidly move on. The COVID Moonshot team is huge. There are over 130 of us and increasing. Although it's invidious, I particularly, because this is a medicinal chemistry meeting, want to highlight the design team I work with. We have an amazing group of folk from UCB who've contributed behind the scenes. There are more people who've contributed some brilliant ideas. The Pastera folk who've been in at the very start setting up this project. Colleagues at Thames Partners, ex-Pfizer folk, including Gwen, who's our pharmacokineticist, Ben and Petter from DNDI, who DNDI is now a proud sponsor of this project. Bobby Glenn from the University of Cambridge, Tatiana, who was one of the instigators of the project from Enamine, Haim, who runs the assays from the Weizmann, contributes to the design team, absolutely vital to our efforts, as is Darren, who provides interpretation on the crystallography. We have been supported by UCB, CDD have provided the databasing that has kept our global team together. We have just started to receive amazing support from Novartis in terms of access to their in vitro ADMET panel, and also Takeda have provided some amazing profiling support. Thank you very much indeed for your time. I'll be available in one of the breakout rooms afterwards if you've got questions. I'm recording this at the end of August. In 2 weeks' time, there will be more data. I hope I can share it with you then. Thank you very much.